Assalamu alaikum, my dear students. Welcome to all of you in grade 11. First of all, congratulations from the core of my heart that you people promoted from grade 10 to grade 11. My name is Adnan Alam and I will teach you biology. The book recommended by the Federal Textbook Board is the National Book Foundation Biology Grade 11. So all of you are advised that please take this book because book is very important. It should be with you. My dear students, these are the online classes and we are going to start our first lecture today, and the first chapter is cell structure and function. First of all, we have to know what is cell, what is the meaning of cell. So my dear students, the word cell meaning is it is a compartment. Cell means compartment, chamber. And in 1665, by Robert Hooke, discovered the cell, who observed in the card bark he took, and he came microscopic study, and he observed small chambers, and these chambers were assigned to be the name is cell. My dear students, cell is the basic, it is the basic unit of structure and function of all living organisms. So in this chapter, we will study the structure of the cell and the function of the cell. In the mid of 19th century, this most popular cell theory was developed by Schleiden and Schwann, and the main points of the cell theory I want to write here. The first point of the cell theory, that the cell is the basic unit of structure and function of all living organisms. Number 2, all living organisms are made of one or more than one cell. And the third and the last point, that new cells arise from pre-existing cells. Cell is the basic unit of structure and function of all living organisms. All living organisms are made up of one or more than one cell, and new cells arise from pre-existing cell. These 3 points, cell theory, have a great importance, which laid the foundation for further study in the cell. My dear students, now we are going to start different techniques. In these techniques, we will study different techniques which are used to study the cell and its different aspects. So now we are going to start these techniques, and the first technique is cell fractionation. So let's start. My dear students, the first technique which we are going to study is cell fractionation. What does it mean by cell fractionation? Cell fractionation means to divide a cell into different fractions. Why we need to use this technique? First of all, we need to learn why we need cell fractionation technique. My dear students, if we study the cell under the microscope, we can study the cell, but still some of the organelles which are present in the cell are not visible to us. So we need to isolate them. For isolation of different type of fractions are the organelles which are present in the cell, we use this technique called cell fractionation. So my dear student, first of all we have to define what is cell fractionation. Cell fractionation, it is a technique to divide the cell, to break the cell into different fraction or components. My dear students, it's a detailed process which I will discuss with you, but let's study its steps first. The first one is homogenization. In the mechanism, first grinding of tissue to make a homogenized mixture. Homogenization, we need a micro grinder which will grind the tissue and it will make a mixture, and then for further homogenization we will apply different type of enzymes to break different compounds. Like we will apply pictase, an enzyme pictase, which will break middle lamina of the cell wall. The middle lamina of the cell wall is usually made up of calcium or magnesium pictate. So it will break the middle lamina. Cellulase, if we use cellulase, an enzyme, cellulase, it will break the cellulose component. So by grinding and applying different type of enzymes, we will get a homogeneous mixture, and then we will centrifuge them, and after centrifugation we will separate different size and density, different size particles and different density level particles. We will separate them, and then after isolation we will study them. Let's study centrifuge first. The centrifugation is a process by which we spin a mixture on different speed, we rotate them, and the machine is used, and that machine is called centrifuge machine. And that centrifuge, let's study the steps. We took tissue which is desired to be for the cell fractionation. We will grind it in a grinder, and then we will take a homogeneous mixture and different type of enzymes we will apply, which will make a homogeneous solution. Then we will spin this homogeneous mixture on different speed in centrifuge machine. My dear students, the speed of the centrifuge machine is usually calculated in RPM, revolution per minute, rotation per minute, and G, gravity. And the gravity value is 9.8. So my dear students, 1000 times gravity, and 1000 times gravity, we will rotate, we will spin this homogeneous solution, and after spinning, we will get some heavy particles will be sedimented on the bottom, and the rest of the solution will be here. Now, two terms you need to learn. This is called pellet, and this solution is called supernatant solution. Supernatant solution. So this is the basic rule that if you are spinning a solution in the low speed, low gravity, then the heavy particles will settle first. So if we rotate or spin this solution with 1000 G, the nucleus and the cell debris, membrane debris, different type of cell debris will be obtained in the pellet, and we will remove this supernatant solution into another test tube, and the nucleus and cell debris will be isolated. If we desire just to study the nucleus, so our process is completed. But if we want to study more organelles, then we have to spin this supernatant, which we transfer to another test tube to the next speed 20,000 G for 20 minutes. First solution time was 10 minutes. It was spun on the speed of 20,000 G, and mitochondria and chloroplast like organelles settle down in the pellet, and we isolate these organelles from the pellet, and the next supernatant solution is transferred to another test tube. If we desire to study the mitochondria and chloroplast, then up to here our process is completed. But if we wish to study other organelles, we will spin this solution with a new speed, that is 80,000 G for 60 minutes, and now we will get plasma membrane and the debris of different internal organelles membranes. If you want to pursue this process, again we will take the supernatant and we will spun, and we will take ribosomes in the pellet 150,000 G for 2 hours. We will get ribosome, because ribosomes are so small particles. My dear students, when we study the ribosome, we say this is 80, this is 90 S ribosome. So what does it mean by S? S means Swedberg unit. Swedberg unit. and it is actually the rate of sedimentation. It is the rate of sedimentation. On which rate it is going to sediment in the pellet, that will be the size of the ribosome. If it have more S, then it means that the rate of sedimentation is high. If less S, the rate of sedimentation is low, means high S, large ribosomes. Small S, small ribosomes, because the small molecules will sediment in the pellet in great period of time due to its small size. Now, this was a density gradient centrifugation. This is the density gradient centrifugation, and by this centrifugation we can isolate different type of cell organelles which can be subjected for further study. Now the next type of the centrifuge is the density gradient centrifugation. Density gradient centrifugation. Once again, giving you a quick review, and then this centrifugation is differential centrifugation. And now we will study the next type of the centrifugation, which is called as density gradient centrifugation. So first, having a quick review, cell fractionation, the definition is here: to divide the cell into different fractions. The mechanism is first grinding of tissues to make a homogenized solution. Homogenization is the process by which we make a homogeneous mixture. For this purpose, we need two mechanisms we use. First is grinding, and also we apply some of the enzymes. These enzymes play a vital role to break the different type of large chemical compounds are present, like pectate, like cellulose. Then centrifugation. In centrifugation, we spin the tissues with different rate, with different speed, and then according to the size of the particle, they settle down. So when they settle down, this area is called pellet, and the solution present above the pellet is called supernatant. These two terms are very important there. Sorry, you must learn. And my dear students, a different spinning on with different speed and different on different time giving different duration, we will obtain different size particles. The smallest size particle, if we want to obtain the smallest size of organelles, if we want to obtain, we need the high centrifugation speed, and duration must be 2 hours. So by this way we completed the centrifugation, but this is not the only type of centrifugation. This is differential centrifugation. Now we will discuss density gradient centrifugation. So let's study. Density gradient centrifugation. First of all, for this type of centrifugation, we need a homogeneous solution. Homogeneous solution we need, and when we get the homogeneous solution, we will centrifuge them, and again from the palette we will isolate the large size particles and the supernatant will be moved to another test tube, but this time this test tube usually the saccharose solution is used here, saccharose solution. So the in saccharose solution, what will happen? The largest particles will be of saccharose will be here, and the low size particles will be here. So when we will transfer this supernatant, which contain different type of organelles, they will mix with saccharose solution and they will start sedimentation. But for example, mitochondria and chloroplast, these type of organelles will stop moving here because here the saccharose particles are present. So we will get the chloroplast and mitochondria, for example, will present here. After that, the plasma membrane, and in the last, the ribosomes will be present here. So then we can separate this part, this part, this part, and it contains different type of the organelles. So these are the two types of centrifugation which are used for the self-fractionation technique. My dear students, hope that you have you got good understanding of today's lecture regarding the self-fractionation. In our coming lecture, inshallah, we will pursue different type of other techniques which are used in cell biology, which help us to study different type of the cell organelles. Today we studied the self-fractionation. Hope that you got good understanding. If still if you have any problem, you can come on Zoom for the live session, question answer session, which should be scheduled and will be shared with you, that on which time we will be available for the question answer session. So I will be happy that you should join the Zoom session also. And if you got any doubt or your concept is not clear, you can ask me frankly. I will appreciate the student if he join me and ask a question. I like that the students ask the question, because more asking, more learning. If you ask more questions, but the question should be related with the topic, and it should be according to the topic, according to the syllabus. So hope that you got good understanding. Please stay more time in home due to COVID-19 epidemic, and stay safe, stay blessed. Thank you so much.